I'd like to welcome you all to another CAIRIBU Arctics Community Forum. And today's topic is pretty cool. It's on how to write a scientific paper. I'd say it's an extremely pertinent topic that I've had on my mind for the last couple of days. My name is Mike Nugent. I'm a postdoc at the University of Texas at Dallas, and I'll be moderating today's session. And today's topic is a really important one, and we have a really great speaker. Although I might be a little bit biased and she's my boss. Today we'll hear from Dr. Nicole Nisco about many of the important aspects in preparing and writing and publishing a scientific paper. So just a quick introduction. Dr. Nisco has a really extensive background in microbiology and host microbe interactions. She did her PhD at MIT with Graham Walker. And then after completing her PhD, she went on to do a postdoc in Kim Orth's lab at UT Southwestern. She's currently assistant professor in the Department of Biological Sciences at the University of Texas at Dallas. And her lab studies many things, but predominantly host microbe interactions and microbial ecologies in the postmenopausal urogenital microbiome. And as someone who's had the pleasure of writing a number of papers with her, I'd say that she has an excellent perspective with an extensive experience in this topic. Before we get started, I'd like to call your attention to the survey for the evaluation for this event that'll be posted in the chat. And then I'd also like to welcome our speaker to the stage, and I'll hand it over to Dr. Nisco, and let's get started.
Hi, thanks, Mike, for that great introduction. Very kind. So today I'm going to talk to you about how to write a scientific paper. And this is a version of a talk that I've given here to our own students at UT Dallas, our graduate students, to try to get them thinking and prepared. And I think it was very helpful. So I'm happy to share this with a larger CAIRIBU community. And I want this session to be very interactive. So if you do have questions at any point, please, raise your hand or type your question in the chat, and there'll be like spots throughout where we can come and address those. But if you have anything really pressing, feel free to speak up. But I want you guys to get the most out of this session as you can. So please feel free to ask me any questions that you would like to, or ask for any clarifications that you need. So again, our topic is how to write a scientific paper. And I think this is one of the most challenging things for us as scientists, because in our training, we're taught how to do experiments, we're taught how to design experiments, we're taught how to analyze data, but how to actually construct that into a narrative is something that we don't necessarily have courses on. I think a lot of programs are starting to do that or have done that already, but I think it's something that I know is challenging for our students here, and it's probably challenging for students across the board, and it's still challenging even at the level of a postdoc or a professor. I think writing is something that's a little more difficult for our analytical mind. So if you struggle with it, don't feel bad about it. Everyone struggles, and your advisor probably also struggles too. So they're just at a different stage than you maybe. To kind of go forward, I'd like to just take a survey of the audience if you struggle with scientific writing. So if you struggle with any form of scientific writing, please go ahead and raise your hand. So we have some people writing in the chat. So I think all of us, probably struggle with this. And would anyone like to share what aspects of scientific writing are difficult for you? You can do it over the chat. And we have both hands raised for Mike, hand raised for Jonathan. Does anyone want to share any aspects of scientific writing that are most difficult for you? So Jacqueline says summarizing information. We have sending drafts for feedback. I think that's something that everyone feels, and I do have a little section in there on that. Getting started. Sam says synthesizing experiments into a coherent narrative, and Jonathan says using the right language. Right. So hopefully I can address some of these challenges that we're seeing in the chat through the seminar. But if I don't address it, please let me know, and I'm happy to offer any help that I can. All right, so the first part of this talk is a roadmap for writing a scientific paper. So it's going to kind of take you through a strategy that I use within my lab for how I encourage my trainees to start writing. And, just to start off, if your advisor has a very specific way that they want papers written, please ignore what I'm saying. But if you are looking for guidance, this is a roadmap that we use. And so when should you start writing? And so that's probably one of the most difficult questions, is when do you actually get on your computer and start your draft? When do you start that Word document or LaTeX or whatever you're using and start that draft? And so does anyone have any ideas, in the audience of like, when do you think in the process of doing your experiment should you start writing? So Chris says write methods right away or as you're working on them. Mike says start writing methods pretty early. Yep, because you should be writing your protocols, of course, as you are doing the experiments. All right, so I think that kind of summarizes what we actually should be doing, is that many scientists feel like they have to have every experiment complete before they should start writing. And that's actually not the case. So you will get done faster, and it will be a better process if you start writing earlier. So you don't have to have every experiment done before you start writing. And this is because the act of organizing your work that you've already done into a manuscript will reveal any missing experiments or logic gaps in your work. So you still might have a couple of experiments left, but that doesn't mean that you can't already start organizing your work into a manuscript, especially if they're things that you're doing to validate or using a second method to confirm something. If you start to organize your manuscript early, then you'll be able to pick out the experiments that you need to do to test your hypotheses more effectively earlier, so you're not going back at the end and redoing a bunch of stuff. So my suggestion is to start writing early, and this isn't just methods. This is actually start writing and organizing the manuscript early, so you can identify gaps before you are up against a deadline. So deadline saying maybe it's time for you to start thinking about defending your thesis. If you already have a good idea of the organization of your manuscript, which will eventually become a thesis chapter, then it will be a lot easier to get started or to actually finish, because you've already gotten started. All right, so what are the, so these are the common components of scientific papers, where you're going to have your title page that will consist of your title, author's affiliations, and then keywords, and sometimes there'll be a running title or word count depending on the journal specifications. There'll be an abstract, and that abstract again will depend on the discipline, what its structure is: introduction, methods, results, discussions, figure and table legends, references, acknowledgements, and author contributions. And so these are the textual parts, and then of course there are your figures, right, and your tables, which are where your data are. And so depending on the journal, the legends will be either inserted into the results section or will be put towards the end of the manuscript. And so whenever you're thinking about starting to organize your work, you might want to identify some target journals that you would be thinking about submitting to, just so that you can be familiar with their requirements and their guidelines. And again, most journals offer copy editing and will format your original files and figure legends into the final PDF version. There are some journals that have actually template documents, or that you might use LaTeX, but most in our field are going to do all this copy editing for you. And so how do you start writing? So if you don't mind, again, to share your ideas in the chat of what's the best way to start writing a manuscript. Where do you start? So so what sections do you think you should start with, for example? Any ideas in the chat? All right, so we have Namrata saying methods, Rishi saying methods and results. Any other ideas? All right, so those are some very good ideas. And so so the way that I like to start is actually preparing the main text and tables. And this is just assuming that you are writing your protocols as you go, so you already have some draft of the methods. But really, where the actual process of writing your paper starts and getting that narrative in place starts is when you're preparing your main text figures and tables, because that's where your data is, right? And so in doing this, of course, you shouldn't forget to write figure and table legends. Those are necessary. And then the second step after we've started a draft, and this is going to be probably draft 1 of, maybe 10 of your figures or tables, you want to organize them so that they follow a logical narrative. So what I like to have my trainees do is make a figure outline. So take your figures that you've made that have your data that you've been working on, and then organize them into an outline, into a narrative that is logical and can answer the hypotheses that you're putting forward in the paper. And it's really important to keep in mind that the best order for your figures and tables may not be follow the chronological order in which the experiments or analyses will be performed. So, a lot of students will organize things just by the date when they did them. But, you may, that's not actually how your paper is going to end up, because the logical flow of information may not be the order in which you did the experiments. And so maybe you went back because you realized that you needed to do this other experiment to show something, or you found something cool. And your narrative will change over time, right? Because your paper is really about the data, and you can try to predict the data, but data is the data, right? And so just remember to be flexible when organizing your figures and tables, and they don't need to follow the chronological order that they were done. And so this exercise is usually really helpful because it's here that you can really start to notice gaps in the logic of your work that need to be filled in with more experiments or analyses. And so when you have all of your figures lined up, you have, okay, this is going to be figure 1 and it will have these panels with these data, and then figure 2 will logically follow from that. You might find that the connection between those two figures, there isn't something that you can easily state in the text, or if your hypothesis is X, then maybe the data that you have and the experiments that you did don't completely address that hypothesis, and you haven't really fully tested the null hypothesis with your experiment, so you might need to add some. And so this is a really useful exercise, and I think I hope my trainees also think it's useful, because it's usually where we start. And so my third step is writing the methods, and this is just because I want to get the logic done, but I can understand why people might want to start with the methods first, because it's the easiest thing to write, because it's the protocols that you've been doing. And so for me, the methods are very important, but again, they are not going to be the actual narrative of the manuscript. And so again, this should be the easiest part. You should have your methods well documented in your lab notebook, and really you should be writing them along the way while doing experiments, and you can always come back and revise, but you don't have to work on the final versions first. I mean, if it helps you to get something on paper, that's fine, but without organizing your figures into a logical order and thinking about the overall outline of your manuscript, it's not going to really matter if you have methods on paper or not, right? And I always suggest that having typed up protocols will help you when it comes to writing the methods, and because you already have it typed up, and you can then summarize it into a methods format. So in general, methods should be sufficiently clear for someone to replicate your work. And that's, really the main reason for reporting them, is so that people can go and replicate your work, or replicate the experiments that you did and applying it to their own system or their own questions. And so this can be tricky when there's certain word limits for things. And so some journals require very detailed methods, like cell press journals have STAR methods where you have to actually use very great detail and include lists of reagents and everything that you've used with catalog numbers. And some will allow supplemental methods, is say like you can't provide enough detail within the word limit that they're giving for the main text, then you can actually write, have in your supplemental methods to provide more detail that would actually allow someone to replicate your work. All right, so then my fourth step in my suggestion is to write the results section. So you have your figures, you have them all in an outline. So you have them in an order that follows a logical flow. You know, you've written some methods, so you feel like you've accomplished something, right? You have words on paper, and now it's time to look at your figures and your tables and all of your data and write the results section. And so in this section, you're going to be describing the data presented in each figure and table in order, right? So so this is why it's important to set the order of your figures first, so that you can make sure that you're describing them in the correct order in the results section. And so in my opinion, it's good practice to report both the effect size and the statistical significance. So many times when people are reporting and describing their data, they'll just say that, oh, there was a significant difference, but they won't report the actual change that they're observing, the effect size. And so in general, for me, that's my suggestion is describing both the effect size and the statistical significance, because effect size will get at more biological significance, right? And this is, you know, a peeve of mine, but, you know, many other reviewers will feel the same. The word significant should only be used in the case where a hypothesis testing is performed, and, you know, and your p-value is less than what you set as your alpha. So, so yeah, so this is just, you know, little tricks for writing. I'm sure people have different opinions, but that's mine. And so when you're writing your results, you want to separate them into sections by figure or by major result. And so this is done with section headings normally. So you're going to have section headings for each figure or major result. Sometimes figures are very large and contain many panels. And so if this is the case, you might have more than one section per figure. And so your section should each convey a major idea. So these section titles should state the major findings of the section. So it should be clear to the reader when they're reading that section that this is what reading about the data and looking at your figure is going to convince them that whatever finding you're presenting is true. And then sometimes there's not going to be a major finding in a section, like maybe you're just describing patient demographic data or something like that, or you're describing the set of genomes that you use in an analysis. Then, you know, the title should be descriptive of the context of the section. So basically, the reader should know what to expect to see in that section when they read that section heading. And so I usually have them as statements. And so while writing the section, you know, you also may realize that you're missing experiments or analyses. And so if this is true, you then will go back and complete them, right? So you'll be like, oh, well, actually, if this is my hypothesis, I have, you know, these data that might support this hypothesis, but this alternative hypothesis might still be true. So I need to do an experiment to address that. Or I need to do an extra analysis. I didn't account for this or that. And so you'll have to go back and do that. And then you'll kind of, you know, go back and start the process again and just make tweaks along the way to your figures and then and so on and so forth. So the idea of, you know, writing a paper is that, you know, it's going to be kind of an evolving process, right? So so what you kind of, the idea that you started off with is probably, and even maybe even the the flow of the figures, the order of the figures that you start off with might not be what you end up on. And so you have to be just flexible and really just follow where the data and your belief and support of that data follow. So so that's writing the results section, which is very important. And then usually I'll organize the supplemental material. So you notice I haven't touched on intro or discussion yet. And this is, I think a lot of people want to start chronologically, and so they'll just start writing the abstract first and then the intro. And that's really just not the best way to do it, because you don't know what you need in those sections until you have the meat of your manuscript complete. And so, so because the supplemental material contains data that you'll be probably talking about in the results section, you should also do this rather early. So when writing your results, you'll identify where supplemental data or tables are needed. And so then you'll need to organize your supplement once you've identified what needs to be included. And you might have that idea already. You might have already kind of identify things as supplemental material when organizing your figures. So again, like when you're organizing your panels in step 2, you might have already kind of said, oh, well, we have this supplemental data that will support this, but isn't completely necessary to include in the main text. So again, supplemental material includes data that is necessary to further support the conclusions drawn in the results section, data that's necessary to demonstrate rigor of your analyses, or data files needed to reproduce analyses. So when you're doing kind of like big data studies, it's common. And then data or files that will enable the use of your data set by others. And so this is kind of usually how I define what should go in the supplement. So large supplemental data files like raw sequencing data usually should be deposited in appropriate public repository. And so you'll just have a link to that in your paper, and you won't necessarily have it, in the supplement itself. And so for most journals, this will be done before submission. So most journals require you to have deposited your data, like in GEO if you're doing RNA-seq, or in and the SRA if you're doing sequencing, genome sequencing and things like that. They'll require that before you've submitted. All right, and so what's next? And so these are kind of interchangeable, and you can really do it in whatever order you want. So now we get to writing the discussion and the introduction. And so, again, these two steps are completely interchangeable in my mind. And so for the discussion, you're really going to be discussing your findings in the context of the existing literature. So, so it might be easier for you to write the introduction first so that you already familiarize yourself with the existing literature, and then writing the discussion might be easier, or vice versa. And so if you have a model figure that you've made, so say you've figured out like a new gene regulatory network or something like that, and you have a model figure, this is a good place to describe it. And then it's really important in discussion to identify study limitations and lines of research that need to be continued. So for the introduction should provide sufficient background so that the reader understands why your study is important, new, and interesting. And so, it's not going to be for at least a scientific paper, a peer-reviewed paper. It's not going to be an entire chapter on the, on the background of this field since, since the 1950s or whatever. It will just be what they need to know to understand why your study needed to be done, basically. And in this section, you should also define your hypotheses and the objectives of the study in the context of what is known again. So this is where you kind of provide rationale for why you did what you did. And then I like to end the introduction by summarizing the most important findings in the final paragraph, and to entice the reader to keep reading into the next section. I don't think everybody does this, but this is something that a lot of people like to do. And so, so then you have your introduction and your discussion. So So additional pointers for writing introduction and discussions is you want to make sure they're well cited. And so this is, usually the first kind of feedback I'll give my students in these sections is that you don't have enough citations, and you need to make sure that every statement of fact is supported by a citation. So for, basic science journals, there's usually not a citation limit. So if you're submitting to one of these, then I really like to make sure each statement of fact is supported by a citation. And so, that can be hard to do when you think something is just general knowledge, but your advisor can help you point that out. For medical journals, I found this tricky because there are citation limits, and I find that strange. So, and maybe this is to prevent not, too much self-citation and stuff like that, but some limits are like 30 or 25. And sometimes I don't really find that sufficient, but these can be tricky to navigate. And so I would really ask your advisor for help with that, especially if they're more used to, publishing in these types of medical or clinical journals. But the thing that I guess helps a little bit in those cases is that usually the overall word limit is much less in medical or clinical journals. So usually the scope of the introduction is also less. And so sometimes it's easier to get away with having less citations. And so again, another pointer is that citation should be for the article where the primary data to support the statement was first published and not review articles. And so especially if you're publishing in a basic science journal where there's not really much of a citation limit, you really should be citing the paper where that finding was made, and not a review that talked about it. Because people want to go back and verify that finding. I do it all the time where I'm reading a statement and I want to go back and I want to find the original paper so I can look at the data myself. And then they cite a review, and then you go down this hole and that review cited a review, and that and then you're just going to this black hole of reviews and you can never find the primary data. So just in general, it's always good practice to cite the paper that actually made the finding. Okay. So, and then in the discussion, it's important to present the limitations of your study. And so many journals actually require this as a separate section within the discussion, and that's great. So in this section, it's very important to clearly convey the limitations of your study. And this is important to prevent overinterpretation, right? So you want to make sure that the readers understand there's limitations to every study, and that is fine, right? I mean, we cannot do everything, and one paper should not do everything. And so the fact that there are limitations is fine, and it's good to just make them transparent and known, and it can identify, areas of new study as well, right? Okay, and so then at the very end is where we get to writing the abstract. So kind of after you've done the intro and then the discussion, then I suggest going back and writing the abstract. And so the abstract is going to contain elements of your intro and your results. So it's really important for you to do that after you've written those sections. And then you can compose the title, because by the end of this writing exercise, when everything is more complete, you know what the major finding of your manuscript actually is, right? And then, selecting keywords and stuff is pretty easy. So the title, again, should accurately convey major findings. I personally believe that cute and funny titles should be reserved for reviews or commentary, but your advisor might have a different opinion, and so that's completely fine. And so abstract format and length depends on the journal. Again, so medical journals, clinical journals will have a formatted abstract with defined sections, and then usually basic science journals will just have a single paragraph around 250 words or something like that. And so then I go and make sure the references are in the correct journal format. So you can download journal specific reference styles and EndNote, Mendeley, etc., whatever you use for whatever your lab uses for citation. It's usually good to all get on the same software just so that you can easily exchange drafts. And then writing the acknowledgements and author contributions. So journals usually have guidelines on how to format those, so you'll want to follow those guidelines. And then acknowledgements are something usually your advisors should take care of because they contain funding information and grant numbers and stuff that they know. So I guess hopefully that was a useful kind of roadmap into how to kind of tackle starting to write a scientific paper. And so now I'm going to go into a little detail about the most important part of a scientific paper and how to organize that. So what's the most important part of the scientific paper? The figures, right? And the tables. And the reason for that is because I was going to have you share your thoughts, but apparently my order is all messed up. That's okay. Your data is the centerpiece of the manuscript. That's what the whole manuscript is about. It's about the data, right? And so without that data, you don't have a manuscript. So your figures, which contain the data, and tables are going to be the most important parts of the manuscript. And so that's what you're going to spend your majority of time really working on, is getting those figures finalized. And so you need to present your data in a way that is accurate, easy to interpret, and accessible. So and so this, this is why you might end up with like a version 10 of a figure, because you might be going through iterations where you can improve upon how you're presenting your data. And it can also help to present data in a way that's visually appealing, right? So you want people to want to read your paper. So you want them to want to look at your figures, right? And so if there's a way that you can use different color palettes or things that are visually appealing, and there's a lot of tools for doing that, then, that can just help with the whole reading experience, right? And again, every conclusion you make in your manuscript needs to be supported by data provided in a figure or table. And so this is really, important. And this is why your figures and tables are really the most important part of your manuscript. So how do you turn your data into a figure? So what software do you use to make figures? So in my lab, we use Adobe Illustrator to compose multi-panel figures. So if we, all of our figures are usually multi-panel, A, B, C, D, etc. And so we use Illustrator to compose these. If you are analyzing and producing images of microscopy data, you can use things like ImageJ or Fiji or Zen Blue if you have a Zeiss, etc. And we use GraphPad Prism to just make plots and all of those kinds of graphs and plots and everything. And then also it can be useful for performing biostatistics. So, they usually, Prism won't sometimes it's very difficult to perform the wrong test in Prism if you put your data incorrectly. And so it can really help you in learning, which statistical tests you should be applying. And then, of course, depending if you have like big data, like microbiome data, for example, you might use R packages to be able to start making some of your figures and plots, especially if you're more computationally inclined. We use BioRender, I know, and then I think there are some other free versions of this that exist now, or ChemDraw for making diagrams, workflows, or chemical models. Software you should not use. And so this is something I believe strongly in, is not using Photoshop, because Photoshop lets you manipulate data and especially images in ways that are not ethical. And you can use it without doing that. But I think for me, it's just easier to tell people to not use it. And that way we don't run into that issue. At least with things like Western blots or microscopy images, etc. So PowerPoint is great for organizing things, but I don't like it to use it to make figures, and that's because it immediately downsamples images that you put in it. And so if you try to like print your PowerPoint slide into like a PDF or make an image file out of it, it will make a raster low DPI graphic. And so it won't actually preserve the resolution of the graphics you put in it. So it's really great for presenting and organizing your data, but for making final versions of figures, I don't like to use PowerPoint. So that brings us to the question of what is a raster graphic? And so and even this image is a bit rasterized. And so my lab often calls me the raster police because I can immediately tell if an image is rasterized or not. And so what it means is that basically if it looks fuzzy. And so what's kind of behind that is that raster artwork is this flattened image consisting of many pixels, and it's not scalable. So that's the key, is that as you enlarge the image, we don't make more pixels, those pixels expand, and so then they make the image fuzzy. And it's not editable. And so what we prefer are usually vector graphics, because those are consist of vectors or points and paths. And so this type of image is scalable, so you can make it bigger and smaller, and it will just increase the number of pixels to do that, and without it getting fuzzy. And so usually, if possible, we like to use vector graphics. And so that's why we use Illustrator, because everything in Illustrator that you make in Illustrator is going to be vector graphic. If you export things as PDFs, those are editable in Illustrator as vector graphics usually. And so that's something you can talk to your advisor about, but just in general, that's an example of why I don't like to use for example, PowerPoint to actually compose a final version of a figure, because it will make that kind of raster graphic. All right, cool. So how do you decide whether to present your data in tabular or graphical format? So that's another point. When you're actually just making your figures and you have data, do you present it as a figure or a table? And so this is an example of a published figure from one of my students' papers. So for data that can be plotted, graphs are usually preferable because they allow you to visualize effect sizes, data distribution, and summary stats. So if we can plot it, because then you can actually more visually see. Sometimes there's just going to be too much data that it will be impossible to plot, and it will be better as a table, or it might be summary data that would make more sense as a table. But visual representations are preferred, at least for me. So tables are useful for reporting summary values, binary or categorical outcomes, or say you have you've done like a scoring where you're saying it's a high, low, medium, or something like that, and it's not, you don't actually have specific data points. That's something that can't easily be plotted. Then you can go ahead and report that as a table. But if it's something, again, that can be plotted and you are looking at differences, then my go-to is always to plot it if you can. And then some journals may require raw data to be supplied as supplemental material. And so this is always good practice. And so if they do require that, obviously you'll have to do it. And if there's a way to provide raw data, I always like to, especially certain types of data like sequencing data and stuff like that. But again, it's just making sure that everything is open and that people can reproduce your findings. And so for this panel image that I showed you, this whole panel was composed in Illustrator. Panel A images were processed in image J, and panel B plots are made in Prism. And then again, this whole figure was composed in Illustrator. So that's just the approach we took for making this. And so using kind of the appropriate tools will make sure that you're reporting things correctly. All right, so what are some good attributes of a good figure? So figures should be easy to interpret without reading the text. And so if you just have the figure and a figure legend, you should be able to tell what that figure is trying to show. So you can do this with having clear labeling and legends within your figure. So if you have a heat map, for example, then you need to have a color key for that heat map so people know what each color means. And then you need to make sure your labels are clear and legible so that it's very easy to understand what you're trying to convey. And in this vein, it's really important to use legible font sizes. So try to make your font sizes as big as possible. I mean, not like comically big, but big enough so that people with older eyes can read them. And a good figure will direct the eye of the reader to the important data. So again, it should be, you just want to make it easy for the reader to understand what you're saying, right? So understand the point that you're making when you are reporting your data in the results section. So you want it to be easy for them to look at the figure and understand that, right? So an example of this is you could use arrowheads to denote important features and images, and then presenting the results of statistical tests directly within the figure instead of just in the text or in the legend, so that it just makes it easy for someone to understand the whole point of the figure just by looking at it. And then a good figure does not mislead the reader, right? So, so we want to make sure that we're reporting the data as it is, and we're not, over interpreting things or making it easy for the reader to draw incorrect conclusions. So a way to, techniques for doing this are to keep scales consistent between graphs. So if we have graphs within the same figure where you're kind of comparing things between those graphs, usually you want to keep the scales, like on the y-axis, for example, to be similar so that you are really comparing the same thing, and it makes it easy. Sometimes, this isn't going to make sense and you're not going to do that, but I'm sure your advisor will be able to point out times where it absolutely does make sense, and you should really keep those consistent. And then when you're selecting images, including your figures, make sure you select representative images, not ones that just support your hypothesis. So, and in this vein, it's a good idea to quantify imaging data when possible to avoid bias, because it's really easy to kind of, even without thinking about it, to biasly select images that you like better, that look a little nicer and that support your data. And it's fine to show these representative images, but if you also quantify things, then you actually have more concrete support, right? And then make sure you've applied the correct statistical tests. And then I, like to, in most cases, show the data distribution. So we will show individual data points if it's not too messy, or use box and whisker plots so that the data distribution is apparent, instead of showing things like SEM, which can actually kind of make the data distribution seem tighter than it really is. All right, so let's look at an example of kind of making a figure. So this is the first draft of a figure from one of my grad students from a paper that is currently on actually bioRxiv right now. And so this was the first draft of her figure. So this paper is about, is this genomics paper looking at efficacious genomes from different sources, the urine, the guts, and the blood. And as part of this, she curated a collection of these genomes from those sources and is describing that collection here in this figure. And so my feedback for her when I saw this first draft of this figure was that the provenance of the urine and efficacious strains was unclear. So one of the things done in this paper was sequencing of a bunch of urinary efficacious strains. And so where those came from was not conveyed at all in this figure. And so that was probably the major point of feedback. The y-axis legend is missing in A. And the color scheme, I thought it could be improved and made more visually pleasing. So, so this was the final draft of the figure and what's currently on bioRxiv. And so the kind of how the feedback was addressed here was that she added panel A, so that the source and host status of these clinically fecalous isolates was clearly identified, and we can see even the number per group is defined. So just being more clear about about where these strains came from, and so this hopefully is something that's easier for the reader to understand and to see. She added the y-axis legend here as number of genomes, which is now panel B, because it was panel A before. And then she made the color scheme more appealing and then unified it throughout the paper, which is just my personal thing that I like to have things also be visually pleasing, because when else do you get to be artistic? Anyway, okay, so and so that's how she identified and she was able to improve the figure based on my feedback. And so the other part of this figure is the figure legend, which now you can see below. And so what are kind of some pointers for making figure legends? So figure legend title, so the title here, should summarize the major finding or overall points of the figure. So for this figure, there wasn't a major finding because we were just describing the collection. And so this is, the title just kind of describes what's in the figure then, which is these clinical cohorts and the isolate collection. And so here, this, like I said, this figure depicts the overall isolate collection used for the genomic analysis. The legend should describe any abbreviations, acronyms, or notations found within the figure that need to be defined. So here we have superscripts A, B, and C, which are present in this panel B graph, are defined in this legend. And also there are citations within the legend from the papers from which these genomes were taken from. And then if applicable, the legend should include the names of statistical tests used to generate p-values. So in this case, there weren't any statistical tests applied because we were just describing the collection. So there was no need to do that. But if you are reporting, p-values in your figure, then it's good to include that statistical test used to generate that p-value within the figure legend. Cool. All right, we're going to skip this. Just in an instance of time. So that was an example of how a figure could be, how we went from one draft to another draft of a figure. And so hopefully this is something you might do with your advisor. Another, decision to make is when a figure should be supplemental. So when a figure should be supplemental is when it's not necessary for understanding the main narrative or major conclusions of the manuscript. But the supplemental figure should be included if it provides additional supporting evidence for these conclusions, or if it's like the sole support for minor findings. So you might have major findings in your manuscript, and then you might have some minor, less important findings. And so those might go in the supplement. The data for those figures might go in the supplement. Something that's often also included in the supplement are negative data. So things that where you did a test and you wanted to see if a mutant had a certain phenotype or something like that, and it didn't. And so I like to include those in the supplement because a lot of people just don't publish negative data. But if you can, then if you have enough room to, if there's sometimes limitations in how many figures you can have in the supplement. But it's a good idea to do that because it will keep other people from trying to do what you did and also not finding anything, right? And so in just the idea of transparency and kind of trying to move things forward, it's good to report negative data when you can. Also, it might be important for refuting a possible alternative hypothesis, right? And then also what can go in the supplement are data needed to reproduce major analyses. And so these can be provided as a supplemental table, or if they're very large files like sequencing data, they'll be deposited in a public repository. All right, so this is an example of a figure from one of our papers. And so this is figure 2. And so for this figure, we actually have supplemental data. And so that's what I'm going to show you next. And I'll explain to you why it was included as supplemental data. So this figure to see here provides overall pangenome breakdown for the entire Ephicalis collection from all isolate sources, but it doesn't show the pangenomes by isolation source. And so instead of including those individual isolate source pangenomes in this main text figure, where it obviously doesn't fit because it's already a full page, very large figure, we went ahead and we made a supplemental figure for it. So we had a supplemental figure S2, which is basically showing this breakdown. And the reason we did that is because there wasn't really much of a difference between these the compositions of the pangenomes when we broke it down by group. And so this is kind of like negative data showing that the pangenomes have overall similar compositions of like core genes and shell genes, etc., across the different isolation groups. And so we just provided it as supplemental material. And so again, the pangenome composition didn't vary much when genomes were binned by isolation source, so that made us think that it should be in the supplement. And so that's an example of what you might be including in the supplement. Cool. So let's move on to supplemental tables. So supplemental tables can either be provided within the supplemental material as a Word table, or if they are sufficiently large, they might be a separate Excel file. So for example, additional summarized clinical metadata or strain lists might be provided as a Word format table. And if it's not in the main text, you might have it as a supplemental table. But if it's a large file, like an intermediate data file or a large metadata file that are in tabular format, these are often supplied as Excel files. So for example, results of big data analysis that are summarized into main text figures. So these analyses that were used to make your main text figures, you might include as a supplemental data file. And so this will be an Excel file normally, or whatever kind of CSV format or whatever the journal asks for. And so also you might include metadata files that are too large to really be readable in a Word table format. But still these supplemental tables must have descriptive titles, legends, and may also be you also will have to describe acronyms or annotations if necessary within those legends. So an example of that, again, from the same paper is this is a supplemental table S3. This table contains all of the metadata from the genomes that were analyzed in this paper. And so it has a descriptive title, it has a legend, and so this is an Excel table because it is an Excel file because it is very large and it wouldn't be very difficult to read as a Word table. And it's going to also contain clearly labeled column headers so that you can understand what everything is. And so the reason that we include this is because this table is needed to reproduce the genome collection described in the study, and it will convey certain very important aspects of the genomes that we used, their source, how they were sequenced, the coverage, so the quality, so some aspects of quality and things like that. And so providing this would make it easier for someone else to replicate our results and easier for the reader to understand the composition of our genome collection. But it's obviously not something that will fit into a main text figure, right? Okay. All right, so concluding remarks for manuscript writing. My main suggestion is to start manuscript writing early. Don't be afraid to make figure panel outlines before all experiments are complete. And you can use placeholders that that will go in the place of experiments that you're still in the process of completing when you're just trying to outline the logical flow, if that makes it easier for you when composing your figure panels. And remember that you're going to go through many revisions before it's final. So even your figures are going to go through many revisions, not just the manuscript, but your figures will as well. You want to clearly communicate with your research mentor about expectations. So again, these are my, the expectations that I work with my trainees, but your mentor might have different expectations. And so you want to communicate with them clearly throughout the process. So the scope of your project might change, for example. So I would discuss expectations or thresholds for when your project might be publishable, right? And so that might depend on the results that you're seeing, etc., or where you are in your PhD. Small things like what word processor do they want you to use, or do they want you to use Word? Do they want you to do LaTeX? What do they want you to do? Do they want you to always track changes? Do they want you not? As a trainee myself, I've got I had my advisor yelled at me about this before. So just make sure that you communicate clearly what those expectations are, and then what programs that they want you to use for your figure preparation. And then also, if they want to see a complete, but definitely it's not going to be final draft, or do they want you to send individual sections as they're complete? So every mentor has a different style. So just try to communicate openly with them and keep those lines of communication open. And then also really important is to not be a perfectionist. So this gets at, Mike's comment about sending your manuscript for comments can be very difficult because you're afraid it's not perfect and you feel embarrassed, or you think that they might look at you negatively if something is wrong. Your draft does not have to be perfect before sharing. It's none of our writing is perfect the first the draft we have. And so it's important to just get the process started, to get that dialogue started between you and your advisor and other authors, and to not try to be too much of a perfectionist, because your mentor is going to probably edit your first draft very heavily, and that's fine. It's normal, and it's not meant to discourage you. It's just part of the writing process. All right, so are there any comments or questions right now? Please raise your hand if you have any comments or questions about what I've presented right now. I do have some more slides about submitting the paper and reviewer feedback, but I'd like to take any comments or questions about what I've talked about first. Anything?
So say there's a totally hypothetical postdoc who is terrible at aligning their figures. Any tips on alignment in figures?
Oh, yeah, I mean, so Illustrator has like a grid function where you can turn on the grid and that can help you align things. Also, it kind of has auto align, so that's another reason I like to use Illustrator. But maybe that postdoc has an advisor that likes to align things for therapeutic reasons, so might not be that bad. Okay, Chris says she found our comments on graphics and figures valuable. Thank you. Well, thank you for listening.
One of the reasons is because your papers, the urobiome papers, all microbiome papers, have so many figures and graphs, so many more than like the science that I have done. So I'm always amazed at the technical, prowess that you guys have, because in simply selecting what to show, and you did comment about that, but that's always one of my problems. You have so much data. You don't have to report it all. You
Yeah,
have to report that which makes a good story. So thank you.
exactly. So the next part of the process is then once you have your manuscript written and you have a final version that both you and your advisor and your co-authors agree on, then it's time to submit it, and then eventually it'll be time to respond to reviews. So in manuscript submission is usually something you don't have to worry about too much because your advisor will handle it since they're the corresponding author. But there are ways you can help. So your advisor will usually have to compose a cover letter to include with the submission. And so I usually will ask my lead author to review my cover letter, since they're the most familiar with the manuscript. And so they can make sure that the major points that I highlighted are accurate and well expressed. Each journal will have a different submission requirement. So your advisor might ask you to update parts of the manuscript to meet them. So I kind of, on submission day, I usually ask my lead author just to be available in case I'm going to need any new versions of documents. A helpful thing to do, and I need to actually start asking my trainees to do this, is to make a separate document with author information, including their full name, email address, and affiliation that can be used to fill in the author fields in the submission. That's probably like one of the, if you have a lot of authors, it can take a very long time to do that, and also to track down everyone's email address. So that's something that first authors, if you want to be kind to your advisor, you might prepare that for them to make it easier for them when they're submitting it. And so during submission, issue you might run into is everything's usually compiled into a PDF, and tables and figures can sometimes get altered or messed up. And so I always check the preview to make sure the conversion's okay, and I'll send it to the lead author too. And then might be where we'll need to get a new version or alter things, because a lot of the times things can get completely altered in this process, and it's very annoying. All right. So after you submit your manuscript will either go out for review, for peer review, or it will be editorially rejected. Chris has a cover letter question. How should your cover letter how much should your cover letter include a hook or rationale for why it should be published? I've heard different views on this. Yeah, I think it depends on the journal. So for the flashier journals, I think you definitely need to get the editor interested, right? So you need to kind of sell it, but obviously not oversell it. So you want to say, to really convey the major findings and what should get them excited about this manuscript. But I think it's important to not to not say anything that's misrepresented or whatever. But yeah, I mean, for those very high impact journals, I think some editors are looking for that. And usually I like to say how in the cover letter, why I think our manuscript is a good fit for that journal. So why it would appeal to the readership of that specific journal, especially for more specific society journals that will have a more defined readership, to just convey why you think it would be interesting to them and why you think it's a good fit. But that's my view on it. I'm sure people have other views. Cool. All right, so editorial rejection means the editor doesn't think your paper fits the scope of their journal, and this happens all the time, and it's no reason to get discouraged. Once it's sent for peer review, 4 decisions are usually possible. It's accepted, you have a minor revision, major revision, or it's just rejected. Some journals have this option called reject but allow resubmission, and so that's basically just major revision. If it's flat out rejected, then you usually have to move on to another journal. So if I get a rejection after I've done a couple revisions, then I will usually rebut. But if it's a flat out rejection after the first set of reviews, unless I think it was super unfair, it's usually just the case where you move on to another journal. But this is something a decision your advisor will probably make. It's very unlikely that your paper will be accepted after the first round of review, so don't feel discouraged if you get asked for revisions. Everybody does. So peer review and revisions are normal and healthy part of the scientific process. And that's why we submit things for peer review, because we're so involved in the project that we might not catch gaps in logic or errors that somebody who is more withdrawn from the project will catch when they're reading it, right? So it's very important. And you might think you conveyed something clearly, but someone that's not actually in your head might not think, or in the project might not think that. And so it's a good practice to have this peer review and to make revisions. It will make the final product better. So when you get these tips for responding to reviews. So when you get reviews, you'll read through them. Usually it's good to read through them with your advisor if possible, and then to come up with a strategy how to address them. So I usually like to read them and then take a couple of days to digest the comments, because you can get up, like it's just natural and human to get upset or angry if you feel like they didn't appreciate your work, or they completely misunderstood it, or whatever. And so if you just take a couple of days to let everything digest, then you will, react more calmly when you're putting together a plan on how to address them. And it's also important to know, to keep in mind that you do not have to do everything the reviewers ask. There are suggestions. And so a good editor will clearly state the experiments they expect to be done, or points that will need to be addressed in a successful revision. And so whenever I've had an editor like this, I've really appreciated it, because then you can really know where to focus your efforts in revising your work, and you feel like there's a very clear dialogue and expectations. But this is not always the case. So in that case, you'll just kind of have to figure it out yourself, or you'll just have to decide what you can do. So if a reviewer asks for an experiment that you think is clearly out of the scope of the paper, you do not have to do it. Your advisor may go and ask the editor, like, hey, we don't think that this experiment makes sense. So what do you think if we don't do it? Some editors will be open to having conversations and some won't, and that's something your advisor will handle. But whenever you're kind of looking at their feedback, try to see the reviewer's perspective when considering which revisions to make and which to refute. Because, we each have our own perspective on everything. Everything's relative to your experience. So really try to get out of your head a little bit and see their perspective when you're deciding whether or not you really need to do something, or if it's just something that won't add to the manuscript or isn't necessary to support the conclusions. And so if you are refuting and not doing experiments or making changes that the authors that the reviewers are asking for, make sure that your arguments for refuting these things are logical, well supported, and communicated in a neutral tone, right? So you want to not have any emotion that's in your response. You want everything to be neutral, and then you want to support your ideas, and you can even support your ideas with citations if necessary. So usually we just paste the reviewer comments into a Word document, and then I'll have my students respond individually to each comment, in a different color to make it easy to distinguish your responses from theirs. So again, making things easy for the person reading it will just make the experience better for everybody. Some journals require a tract changes version of the manuscript, so just be aware of that, and your advisor should hopefully tell you if that's the case. You will usually in this response reviews document, you want to indicate the textual changes made in response to the reviews, and then provide line numbers of where those changes can be found. And then I like to provide supporting citations if I'm refuting textual changes. So if they're making me change some language and I just don't agree with it, and I think there's support in the literature for the language that I'm using or the point that I'm making, then you can use citations to actually refute those changes. But again, be neutral, try not to be too combative, just be logical. If a reviewer's request is unclear, so this happens a lot where you're just not sure what they're asking, you can try to clarify with the editor, or if the editor is not willing to have that conversation with you, then you can paraphrase what you think they're asking and then politely respond to what you think they're asking, and then in the next round of review maybe they'll clarify. But your advisor might be able to go to the editor and ask for clarification. And so it's good to thank the reviewers for their suggestions. I mean, they do put a lot of time. I know I review manuscripts, put a lot of time and effort into reviewing your manuscript. But don't be like overly saccharine in your responses. So just be like a normal person. So you can thank them, but you don't have to be like profusely, thanking them all the time. You just want to be clear and to the point and logical in your responses. And that's my suggestion. So there's an example response actually. So the review of one of our articles that we publish is actually public on Access Microbiology. And so there's a link here if you want to look at it. But here are some examples of kind of how we did a response to review document in our response to reviewers. So the reviewers, their feedback was in black and our response was in blue. So in this case, they were asking us to use a different database to define antibiotic resistance genes. And so that was easy enough for us to do. So we did that. And we just said that we did it, and then we showed where the results were, the new results were, and where we refined the methods to reflect the new analysis that we did. So again, if it's something that's easy to do and it makes sense, I usually just will do it. This was something where we actually refuted and we didn't do what they asked. So they were asking us to do a whole new analysis. And they even said, I'm sorry that this creates a relatively unpleasant amount of work. And so we just decided that, that was out of the scope of this work, and the graduate student was, about to start thinking about graduating. So it was just not something that we thought was necessary for this paper, and would probably comprise an additional paper. And we didn't think we had enough genomes to do this analysis. And so we just, politely refuted, and then made revisions to take out words that were, again, so they weren't happy with our statistical analysis and the cutoffs that we used. So we just removed the word significant. We left in nominal p-values and let readers make decisions on what they think was important or not. And again, because the point of the paper wasn't really to do a really in-depth analysis. It was just to show these genomes that we had constructed, and that there's a second chromosome for these species of E. raphinosis. So again, you don't have to do everything they ask, and this was fine. The reviewer was fine with it, and the paper was accepted. So yeah, so just talk with your advisor about it, and try to go through things logically and calmly. But yeah, so that's it now. That's it. That's all I have. And if there's any questions about that last section, I'd be happy to take them. And otherwise, I thank you for listening.